Paul, I have a few questions for you. So first up, how does Beam's proprietary LNP formulation differ from other LNP approaches for in vivo gene delivery?
Well, we've done a lot of internal development and discovery of novel lipids and novel lipid formulations to enable delivery of therapeutics to specific tissues that we're targeting. And that involves, as I sort of implied, not just the formulation of sort of standard LNP components, but also functionalizing those LNPs with targeting moieties on the surface. And the combination of proprietary targeting approaches as well as optimizing that lipid formulation is what gives Beam its, I think, differentiated approach to in vivo delivery.
Gotcha. Now, what would you say are the specific advantages that base editing has over other gene editing approaches when combined with non-viral delivery methods?
Yeah, so one really cool thing about base editing is that, again, because there are no double-strand breaks, it's very easy to package together multiple guide RNAs, which are going to target to different places in the genome and perform edits in multiple places in parallel. So you can multiplex together the edits that you'd like to make in a single LNP at the same time. And that's possible because without a double-strand break, you don't have to worry about cutting the genome in multiple places, and then you have chromosomal rearrangements and other undesired safety liabilities from that approach. Base editing gives you the ability to make those edits in a single base pair without cutting that DNA, and therefore it's much more productive and as well as safe to do multiplex editing.
Gotcha. Okay. We had a few questions come in at the registration process from our audience, and for example, someone asked, how is Beam addressing the challenge of achieving therapeutic levels of editing in target tissues using non-viral delivery systems?
Yeah, so getting to a therapeutic level of editing is something that is context dependent in any particular program. It depends on the tissue you're getting to. It depends on the type of edit that you're making, and there's a range of them in our pipeline. It starts with designing a very potent base editor itself, and we have a tremendous team that does a lot of molecular engineering to optimize the potency and continue to advance our technology, like the core of our technology. Another piece is designing the guide and selecting the sequences that are going to be specific to your on-target site and minimizing off-target sites. A lot of work goes into ensuring that you have a very specific guide and screening the genome through a fairly elaborate process of assays and computational approaches to ensure that we don't create any off-target liabilities for the therapeutic. That's the beginning, but it's equally important to think about how you're going to deliver, which is the topic of this session, obviously, these components. And it goes there, they work together. You need to have a highly efficient base editor and guide pair, but then you also need to have if it's an in vivo approach, we're largely leveraging LNPs. If it's an ex vivo approach, we use electroporation or LNPs, depending on the situation. And we do a lot of optimizing of those delivery technologies in order to get the payload to the site of interest.
Gotcha. Okay. One more question for you, Paul, before we move on to our discussion around NK cells. What potential advantages does Beam see in using non-viral approaches for delivering base editors compared to viral vector approaches?
Well, I think the obvious one is the risk of insertional mutagenesis for the viral vector approaches. I think the other piece of this is that they're a little bit different, I would say, types of vehicles. The viral vectors are often to deliver longer DNA sequence or RNA sequences, whatever the case may be. You're trying to insert potentially a full gene, and you're talking about kilobase pair links in a lot of the viral vector delivery approaches. For what we're doing with base editing, we can deliver much smaller RNA molecules, and that's not really suitable. Viral vector is not really suitable for that. LNPs are a much better approach. So I think that in the case of when we're doing base editing, LNPs have both the improvement of safety and also they're suited for the payload that we have in a way that I don't think viral vectors are.